Okay, so we're recording.
Yeah. Yeah, well, thank you for the introduction and the opportunity for me to be here today. Yes, I'm a neuroradiology fellow at the University of Toronto, and I happen to be here this 3 months, so it's nice to be here in person and to give a talk like this. We do have some intellectual property relevant to the content that we'll present today. And I first always like to acknowledge, instead of at the end, you know, the team that worked with me on this. It wasn't all my work. It was the work of many people and the ideas of many people, mainly from UHM. Just for those of you less familiar with the abbreviations today, so DBS stands for deep brain stimulation, fMRI for functional magnetic resonance imaging, Ph.D. for Parkinson's disease, STN for the subthalamic nucleus, which is a small structure of the brain. And at present, there's more than 230,000 patients worldwide who have been implanted with DBS since about the early 1990s. Parkinson's disease is the most common indication, but pretty much every disease has been tried with DBS, from psychiatric disorder to Alzheimer's disease to pain, everything. And then, much like a sort of a cardiac pacemaker, DBS can be, I guess, thought as a brain pacemaker. So there's usually 3 components. There's electrodes which are inserted in the head in a precise target. And then there's usually a battery or a pulse generator that's really over the chest area under the skin. And there's a tunneled subcutaneous or under the skin wire that connects both of them. And the batteries last, you know, from years to many years, depending on the current that's being used. And then, so if we look at the number of publications that are relevant to DBS over the years, you can see that it's probably started around the 1990s when the first STN DBS for Parkinson's disease were sort of becoming mainstream. And as the FDA or other regulatory agencies gave approval for other indications, then the numbers readily increased up to about 2015, when there's new novel leads with novel stimulation that appeared on the market, which triggered another peak in publications. And if we look this time as, I guess, what's coming, or the clinical trials that are relevant to DBS, you can see that most of the trials that are being undergone right now are related to movement disorder, but also psychiatric disorders and cognitive disorders. And a fair percentage of them, about 15%, have a primary outcome related to imaging. So imaging is quite centered to the DBS field and to most trials. Now, today I tried to sort of theoretically split the DBS MRI research into 2 categories. There's going to be a retrospective analysis, which involves electrode position, volume of tissue activated, and normative connectomics. And there's what I call a prospective in vivo fMRI, which allows to look at fMRI changes with DBS stimulation, and also how it can be used to program the DBS, and what are some of the technical challenges associated with this. And I will finish with a few words at the end. So now, starting with the retrospective analysis. So a lot of the research, actually most of the imaging research with DBS for the last 3-4 years has been done this way, mainly because it's easy. Well, it's not easy, but the data required is easily obtainable. So to do this research, you need a preoperative imaging, usually it's an MRI. And then you need a postoperative imaging, which can be an MRI or a CT that shows the electrodes. And the first step is to co-registering those 2 images with the linear registration. So on the left image here, you can see there's a CT with the electrodes in the background. And the red outline shows the preoperative MRI, and they're both overlapping each other. Then the next step is to take your preoperative MRI and normalizing it to a template with a nonlinear registration. And that allows to account for a slight difference in brain morphology across patients. And now the next step would then be to take your CT and your electrode, which are now in the same space as your preoperative imaging, and apply the same transform that you just obtained to move it to a template space. And most commonly, that would be the MNI, or Montreal Neurological Brain, that most people use. And then once it's done, then there's a slight extra step when you do DBS research, because often the postoperative imaging is right after surgery, and there's postoperative changes with like air going around usually the frontal lobes. So most of the available pipeline, they will include a so-called brain shift corrections, which is essentially a few more linear registrations that emphasizes the deep structure of the brain to remove this brain shift that's added from the air going in during the surgery. And then once all that is done, then you can localize your electrodes based on the artifact, whether it's a CT artifact or an MRI artifact. And then you localize them on the long axis and the short axis. And then you can apply all the same transforms that you've been acquiring and move them to MNI or template space. And you can see it gives nice little 3D figure on the bottom right. And in MNI space, there's a lot of atlases available, and you can show your electrodes in relation to different nuclei that are in the vicinity of it. And then the next step so you can either stop there and say, for example, you have clinical outcomes, you can look at certain outcomes and the position of the electrode and make a map of where the best electrodes were. So it would be one option. But most of the field now is doing what's called volume of tissue activated, which is supposed to be a theoretical approximation of where the axons are being activated by the electricity that's being delivered. And there's different models. There's simple model to more sophisticated model. Generally, most of the model, they'll divide the tissue around the electrode into gray, white, and CSF, and assume based on a thresholded impedance, and it will make this sphere that's supposed to represent the tissue that's being activated. Now, there are more sophisticated models, which will involve acquiring like diffusion imaging to refine your impedance around the electrodes, but by and large, most people use a simple heuristic model. And then you can imagine that once you have this VTA of all your patients in the same space, in a template space, then you can do any sort of a statistical analysis and say, this VTA was giving this patient 60% improvement, this VTA 40%, and so on. And so you can do statistics per voxel or per regions and define your so-called sweet spot or cold spot, depending if you're looking at improvement or adverse effects, for example. And then you can relate that to different structure and white matter and gray matter nuclei that you can overlay with some atlases. And then probably in the last also 4 or 5 years, the additional step that's being done is with the advent of these public database, a large public database of healthy people, for example, the Human Connectome Project, which has 1,000 of healthy resting state networks, or there's other data bank which have white matter imaging. So what you can do is you can make a so-called template, and then you can seed the VTA into these templates. And for example, ask, with this VTA, what is the correlation of the whole brain bold signal on the resting state, and see which regions on the brain correlate with that VTA or anti-correlate. And then with that, since you just see you have 100 VTA, then you can show what are the networks in quotation mark that are most associated with improvements or, for example, side effects. And similarly, you can do the same thing. Which tracks were generally associated with clinical improvements or side effects? So that's usually the whole steps that the field has been doing in the last 4 or 5 years. And so I'm going to show you 2 examples that we've done at Toronto Western. There's many others in the literature. So one of them is we looked at the last 20 years of DBS that were done at UHN for the main cohort of patients. So PDSTN for Parkinson's disease implanted a subthalamic nucleus. GPI is a second target, which is the globus pallidus. Dystonia is also implanted the globus pallidus, and tremor in the thalamus, which is a small nuclei called the VIM, and psychiatric patient, which is a subcallosal cingulate area in the frontal brain. And we did the same thing that I was just describing you, and we localized all the electrodes and normalized all to MNI space. I think we had about 500 or 600 patients. And then what we then did is we did voxel-wise analysis and define what were the hot spot, which are shown in warm color, and then the cold spot, which are shown in cool colors. And then we overlaid them on the expected nuclei. It's a bit hard to see here. And so the idea with this is you can then say, even though the subthalamic nucleus is supposed to be the region to stimulate, but with this, for example, we find that it's more the dorsal and lateral aspect, which corresponds to the motor areas that should be stimulated for optimal improvements. So then the idea is you can leverage all the data from the last 20 years to understand who responded better and who did not do as well. So that's one way to do this. Now, the next step is like I was telling you, so then you can seed all the VTAs from all these patients into normative LC data set. On the left side, there would be a functional data set, and on the right side would be a structural data set. And you can see which areas on the brain were most associated with improvement, and then which area on the surface of the brain were connected to the white matter streamline or track that were also associated with improvements. So I guess this extra step allows us to better understand from, as opposed to only a local point of view around the electrodes, of a more global whole brain point of view of what's happening. And I know some of you might wonder, but what about we're using healthy young people to do this, which is a fair limitation and critique. There's a few papers in probably in 2016, 17, when this started, that showed that generally speaking, if you use a data sets of Parkinson's disease versus the HCP, you get similar results, mainly because the HCP is very well acquired, very good data, and you have a lot of them. Was if you try to make your own Parkinson's disease sort of cohort, most likely your data will not be as good, more motion, and you won't have as many. So it's a give or take. You know, nobody really knows what's better right now, but this has been shown to be sort of equivalent in a way. But of course, if we had, you know, a template of 10,000 Parkinson's disease patients, that would be better. Now, the other questions we ask is, so what I was showing you earlier was the global improvement of Parkinson's disease with stimulation. But then we wonder whether we could map out the different symptoms of Parkinson's disease. So we look at the subscore of the global scale in our cohort, and then we map the main symptoms of Parkinson's. So you can see each row in the middle corresponds to a symptom of cardinal symptoms of Parkinson's disease. And the green spot is where you want to stimulate to get the best efficacy. And so this kind of showed us that, you know, someone with tremor, you might want to stimulate slightly higher up compared to someone with rigidity. And so that sort of opened the door to, you know, personalized stimulation in a way, as if you have different kinds of Parkinson, you should probably stimulate in slightly different places. And then we validated this with an external cohort from Berlin, and we use a support vector machine that just classified very rudimentary into quartile of efficacy, and it was shown to be replicated in the Berlin cohort. So what are I guess the advantages of this approach is, as I was saying, you can leverage all the data, all the DBS since the, you know, the early days. So you can inform from previous patients to hopefully try to improve the next patients. The other advantages is we improve our understanding of like what are the circuit or underpinnings of DBS, what are the relevant structures to stimulate to make sure we have the right efficacy. But there are some problems. Is most of the variance, generally speaking, is about 40% that's explained with this approach. So yes, location matters, but there's more than location. So it's hard to apply to a single patient when you only explain 40% of the variance. It's usually, you need more than that to apply it for the next patient that's coming. And the problem with this normalization part of it is that you lose the intrinsic brain heterogeneity that we all have across our brains. And that probably explains why if you stimulate exactly the same place in two patients, you might not get the same efficacy because the circuits might not be exactly the same and the brains are not the same. So you should not expect the same efficacy in everybody. And then the other big problem for me is this VTA modeling, which is at best an estimation. There's a lot of controversy about this, and I doubt that the actual answer is this nice round ball of efficacy around the electrode. So that's another problem. So now switching gear, another project that I think we started at UHN was what I call the prospective in vivo fMRI. And in other words, it's acquiring concurrent fMRI when the DBS is turned on in the scanner, fully implanted. So for those of you less familiar, fMRI stands for functional magnetic resonance imaging. It measures the Bowel signal or blood oxygen level dependent. And for simplistic explanations here, we're gonna, you know, refer it as a measure of indirect measure of brain activity. And it's usually displayed on the brain with warm and cool colors, and showing where the Bowel signal increases and where the Bowel signal decreases. So now if we look at the number of DBS publications that specifically kind of did this technique of concurrent fMRI and DBS, you can see it's a fairly new thing since 2015, where the amount of papers published rapidly increased. And it's only very recent that there are three 3 Tesla studies, which was a major limitation initially when you do fMRI on 1.5. And this was mainly due because most of the model up until very recently were only 1.5 Tesla conditional. Now newer models are 3 Tesla conditional. And if we look at the breakdown on the indication that are being done, obviously most of the studies are done with Parkinson's disease and with the subthalamic nucleus as a target. But there's a lot of other indications which have been tried, such as essential tremor or ET, epilepsy, obsessive compulsive disorder, OCD, pain. So it's a huge field that you can explore with fMRI. Now, what are some fMRI changes that we can observe with PD? So actually, this technique is actually quite, significantly older than what I let it out to be. So one of the first paper was in 2001. And that was before the FDA warning about doing DBS and MRI. That's why this happened. And it's actually quite interesting because you can see here, this one of the first example where each column is a patient with a Parkinson and an electrode in the subthalamic nucleus. And you can see that the blob of colors or the change in bone signal are actually in motor relevant areas like the thalamus, the cortex. So that was, I guess, one of the first proof that the fMRI makes sense. You're getting change in where you expect changes, grossly expect changes in motor areas. Then only in 2003, 2 years later, so this was a single patient. So it's one patient, each row is the same patient. And on the top row, you can see that when the right electrode is turned on, the patient somehow had depressive symptoms. And when the left electrode is turned on, which is the bottom row, the patient had motor improvement. And what's interesting is the bottom row shows what sort of you would expect, right? Like change in bone signal in motor relevant area, supplementary motor area, thalamus. Whereas the top electrode, which was the right electrode, shows completely different area, more like frontal cortices and, I guess, more mood-related region. And when the right electrode was actually replaced in a more correct place, then both electrodes created improvement. So I think this was another very nice sort of example where actually fMRI makes sense, right? The first electrode, you would not expect motor-related areas, and the right electrode, you would expect motor areas, which is what it showed. So I guess circuit-specific engagement demonstrated by fMRI. And there's a variety of other papers out there. One of them is nice to explore mechanistic underpinning of therapies. So for Parkinson, there's a medical therapy and a surgical therapy. Surgical being DBS, medical being levodopa in most patients. And these investigators here, what they've done is they acquired fMRI in patients with medical treatment and patients with DBS. And then they did fMRI analysis, which in this case, I think was a subtraction. And they showed where the fMRI is different across both. So this allows you to explore two therapies that both provide motor benefits, but are relying on a different mechanistic underpinning. One is surgical, one is medical. So that's, I guess, one application of the fMRI. And then another exercise that we did was we looked at all these studies that did fMRI with DBS in the scanner, and we sort of mapped where the investigators reported acute changes in the brain in Parkinson. And what's interesting is the top left image, you can see that most of the colors are actually in the deep structures of the brain, with a few in the cerebellum, with a few in the superficial cortices. And then on the right image, we did the same, but this time with molecular imaging, which is usually PET and SPECT, and with change in metabolic activity. And you can see that the pattern is, I guess, relatively the same. So it's mainly deep structures with the cerebellum and some cortical structures. And what's interesting is with the PET and SPECT literature, there was more data because it's been done for a longer time than fMRI. And in that literature, there's actually investigators that look at long-term changes of DBS. So they would do a PET after surgery and a PET, let's say 2 years after surgery, and they would look at where the PET changes are. And you can see that compared to the acute changes, that's on the left image, the chronic changes observed with PET are markedly different. It almost spares the deep structures, but is much more cortical in a pattern. So while all the fMRI literature right now demonstrates acute effects, there's definitely a value in trying to bring back those patients and try to see what happens to their brain over time to help us understand the disease, but also to see maybe why they have a change or weaning in effect of their therapy. So now how can we use fMRI to program the DBS? So DBS programming is So you have to imagine that these patients are selected first, then they do the surgery, and then they're usually going to go home for about 2 months to let the brain settle down. And then after 2 months, they're going to go, if it's Parkinson, the neurologist's office, and then they're going to go there every week for the first month, then probably every 2-3 weeks for the first 3 months, for the next 3 months, and every month for a whole year. So it's a very cumbersome process. And these patients are usually not the most mobile, and they're quite elderly. They might come from far away. So it's not the easiest sort of post-operative care that's out there. And so while they always go at these visits, the idea is to maximize the benefits while minimizing the adverse effects. So you adjust the stimulation to essentially make the clinical outcome the best, assuming the electrode is in the right place. And then with Parkinson, it's good because there's a clinical feedback. So, you program, you turn on the voltage or decrease the voltage, and the tremor stops. But there's other disorders, for example, dystonia or depression, where there's no feedback. So the patient has to go home for 2-3 weeks, and then they come back, and then they see if it worked or not. So that process is even longer and probably much more error-prone in other disorders than Parkinson. And so the idea here was, can we replace these clinically driven or empirical programming algorithms that have been developed over the years by human observation? And can we replace them and find an objective marker of response to DBS? And for that, at the time, we picked fMRI for no good reason. We could have picked other things like infrared light. We could have picked MEG. At the time, we picked fMRI. And then the idea was to identify clinically beneficial brain activity pattern, and that would then serve as a proxy for DBS efficacy. But we had some technical challenges at the beginning when doing this, as you can imagine. The first one was safety concern. So whenever you have an active implant, the patients can't just go jump in the MRI. Generally, these implants are not, they're either conditional or they're just not eligible. But DBS is MRI conditional, and most of the vendors have somewhat similar guidelines. Most of them, at the time when we did this, were all 1.5 Tesla only. And there's also a limited amount of energy that can be used, which is defined by the vendors by SAR or B1RMS. So usually that window is pretty low, but it's good enough to do an fMRI, which doesn't need a lot of that RF energy. So you can do an fMRI within the guidelines of those companies. And then more recently, as I was saying, is that there are some models which are actually 3T conditional. And that means you can do an fMRI on label with these new models, except there's still one problem, although it's much smaller than it used to be, is these companies usually want the DBS turned off during the scanning, not turned on. There's a slight nuance to this, but generally that's the rule. So if you want to do this experiment, you still need an RAB and some safety testing to show that turning on doesn't create any damage, which it doesn't, but that's the last sort of off-label aspect of doing these kind of things. And then what we did first is we look at the literature when we did that, and we sort of reviewed it and sort of tried to assemble all the risk factors into 2 categories, intrinsic and extrinsic factors that would affect the safety of scanning these people. And so intrinsic factors would be things like, what's the DBS model being used? What's the brand? And then what's the configuration? Is there 2 leads, 4 leads, 2 battery, 1 battery, etc.? And then how is it positioned? So can the patient lie straight in the scanner, or is he crooked such that he has to lie on his side? And then extrinsic factors would be like, what kind of MRI is it? What field strength, what software is on the machine? What coil is being used? And what's the amount of RF energy that's being delivered? So all this complex interplay of factors will affect how safe your scanning is. And the main risk that remains when scanning this patient is actually heating at the electrode tip. So it's not that the electrode will move, like grossly move in the head. It's not that the DBS would turn on and off by itself. It's really heating, and there's a very small amount of induced current at the tip that could happen. But given what's being used clinically, it would be a subthreshold stimulation. So really the main risk is heating at the electrode tip causing brain damage. And so once we had all of this reviewed, then we made this algorithm on how to sort of handle DBS patient that would come to the MRI. And the first thing would be to confirm the DBS device, brand, and model. and then interrogate the impedance, make sure there's no open or short circuit, and then assess whether the DBS can be turned off without too much shaking or tremor happening, and so on. And then the patient will then develop into clinical or research patient. So if they're clinical patients, then we review the guidelines of the company, what scanner we're going to use, what coil, and make sure we're all on label. And after the DBS, we make sure the patient doesn't have any adverse events and ensure that the impedance are stable again, and so on. And then if the patient is research, I guess the main difference is we always have to ask, is the benefit of this patient participating in our research outweighing the risk? Will this benefit to other people or the patient himself? And if we deem this risk to be minimal, then the patient would then undergo the research with us, and then we would adapt our MRI protocol as safe as possible for the patient. So we would use an fMRI that delivers as low as possible RF energy, and then obviously obtain informed consent, ensure there's no broken hardware, scan the patients, and monitor for side effects after. And then, so when we adapted our DBS protocol at the beginning, we used these phantoms. So on the top left is one of the earlier phantom that we use, which essentially attempts at replicating a patient's with a DBS. It's filled with gel, and then there's a few things you can do with it. One of the things we were doing is measuring the temperature rise at the electrode tip when running the scanner. And on the bottom right, I guess, is an evolved version actually from Sunnybrook, an anthropomorphic phantom that also attempts at replicating a patient with DBS. And you can see here, I think there's a small thermometer beside the lead that will measure temperature, and you can assess your sequence. And then one of the questions some of you might have is, what about the artifact or the susceptibility artifact from the metal on an fMRI sequence or GR-API sequence? And actually, it's not as bad as you would think. So when we measured the average of all of our patients of the artifact, you can see that the electrode artifact, or the diameter, was 9 millimeter. And then there's also a coil of wire on top of the head that's also concerned with the artifact. And it only obscures, I think it was, we had 1.9% of the intracranial volume and only about 1% of the cortex on average. So yes, it is a limitation. Yes, you're not going to be able to look at right around the electrode, and you might lose some of the lateral motor cortex sometimes, but it's not prohibitive from doing an analysis. Most of the brain is available, and most of the brain is good enough to obtain a signal. And that was at the numbers I was mostly at 3T with an average of 1.5, but the 3T number were pretty close to the numbers I gave you. Then another concern that we had at the beginning was, is our signal, I guess, reproducible? So if we scan the same patients 30 minutes apart, are we getting a similar, I guess, bowl map? And in this paper here, that's what we showed, is that the strongest areas of both signal change are preserved if we scan the same patients 30 minutes apart. So this was reassuring to us that the amount of signal we're getting is at least good enough to be reproducible. So now if we talk about Parkinson's disease and fMRI. So this was one of the experiments we did at the beginning. Is on the top left image, you can see a schematic representation of Parkinson's disease patient with 2 electrodes in the subthalamic nucleus. And the electrodes at the time were using at 4 contacts. So that's the 4 C's, and each of them can be used to deliver the electricity. And they're spaced by, I think it was 3 millimeter in between each of them. And on the left of the image, there's also a sort of a programmer which shows you which parameters can be adjusted when you program the patient. So you can change the contact that's used to deliver the electricity. You can change the frequency of the current, of the voltage, or the pulse width. So that's usually the main 4 parameters that the neurologist will play with to adjust the electricity. And on the panel B, you can see a coronal T1 MRI of the brain with the 2 electrodes of a patient. And then panel C is a summary of our experimental design. So what we did is the patient will lie in the scanner, and then we would turn on the DBS, and it would cycle on and off with 30-second blocks, or I think it was 6.5 minutes at the time. And then we would do this for every contact, and then for different voltages. And the key here is that we knew from the neurologists which of those were optimal or clinically optimal, because these patients were about 1 year after surgery. So we knew which contact was clinically optimal and therefore our gold standard when we did this. And we did this and we had about 200 sessions of fMRI across, I think it was 50 patients. And then our imaging processing steps were pretty standard. Actually, they were probably suboptimal because our scanner at the time at Toronto Western could not even do a field map. So this could be much better, but this just to show you that with rudimentary equipment and data, it still works. So you can see that you first do motion corrections, you co-register to your T1, register to a template, and then do like regular fMRI statistical analysis with the block design in this case, which was stimulation on and off. And then you can see here it's written 16 ROIs. So at the time, initially when we did this, is I just looked at the first, I think it was 20 patients. And I could tell with my own eyes which of those contact was the best one, just by usually the motor cortex was very strong, the cerebellum and the thalamus. So then we reviewed the literature, as I was showing you earlier, with the PET and fMRI activation across all the publications. And we defined those 16 ROIs across the brain, which were mostly motor relevant areas, as well as some areas we thought were involved in adverse effects. So sure, you might want to have a lot of the good areas, but you also might not want, a lot of the bad area, because you don't want side effects. So that was a very sort of subjective approach when we did this, but that was the best we could come down to when we started this. So the brain was divided across 16 ROIs, and we had t-values of both signal across all these 16 ROIs. And then what we did after this is we divided our data into train and test data. And then once again, like the idea when we started this was not to be any fancy, or just to see, does it have some clinical value? Does it even work? So what we did is we extracted the t-value from the 16 ROIs and only used a linear discriminant analysis modeling. And then that's how we train our model. And the idea was, can the model say, is this a good pattern or is this a bad pattern, right? Because that was our gold standard. It was this defined by the neurologist as optimal, or was this defined as something else that's not optimal? And then we used the same thing with the test data and fed it into the model and say if the model could predict whether that test data was a good one or a bad one. And so here you can see it's an example of two patients. So A is one patient in which we varied the contact that was used to deliver the electricity, and outlined in green is the optimal contact. So you can see that that's usually what we see is there's motor cortex bold decrease, there's cerebellum bold decrease, and thalamic increase. And when we did this, we did this with only left-sided stimulation because we wanted sort of to keep the contralateral brain as a control, and also because the artifact at our institution is on the right side at the top of the brain. So you can see that this pattern, as the contacts being used is moved away from it, this pattern is lost. That would be the middle and bottom row. And now if we do the same thing with the patient in B, you can see that at optimal voltage, there's a similar pattern. But then as the voltage becomes sub-therapeutic or lower, which would be the row above, the pattern is kept, but with lower t-values or lower magnitude of bold changes. And as the voltage gets supra-therapeutic, or high voltage, you can see the t-value goes up, but there's also a lot of spillover in other areas throughout the brain. And so this led us to think that, we can use fMRI to say, what is the optimal, when the optimal colors are there, it corresponds to optimal clinical efficacy. And then when we did this initially, our test data was 39 patients. And when we used obviously more data, which was the contact patients and the voltage patient, we got an accuracy of 88% at predicting the optimal pattern of colors. And then we had two unseen data sets of 9 patients each. The first data set was similar to our 39. So these patients were already programmed. We already knew the answer, and we fed them into the machine learning model. And then, as you can see here, that the accuracy went just, I think it was 81%. So we decreased our efficacy a little bit, but we still maintain above 80%. And then our second data set was the ones we call stimulation naive. So these people were 2 months after surgery. They were just going at the neurologist at that point. And our goal was to say, can we predict what the neurologist will pick in 10 months from now, but 10 months earlier? And you can see that using this data, I think our efficacy at the time was 76% at predicting the optimal pattern. So this was kind of like 10 months ahead of the neurologist we were correct 76% of the time. And so based on this, now we have a trial where the patients undergo surgery. And then about 2 months after the surgery, around their first visit to the clinical neurologist's office, they do the fMRI, then the contacts are screened, then we define what's the optimal contact. And then similarly, the voltage are screened, and we define what's the optimal voltage. And then they go with this at the neurologist's office. And then the neurologist has additional data for their first visit. And the idea here is, can we decrease the number of visits required for optimization, and therefore decrease costs and time and money and effort for everybody? And then also, can we maybe improve the ceiling of efficacy, which is already quite good for Parkinson, but can we make it better? And we've also now that we have more data, we've also now using a whole brain approach, not a 16 ROIs approach. Whole brain, and also as opposed to a supervised method, we're now using unsupervised AI modeling, and our efficacy is now way above 95% when we use these, I guess, fancier number approach. And this is quite timely, because this idea of having a rapid biomarker of success, because you can see that in the top left image, the left electrode is what's been there for the last 10, 15 years, and it's the one we've been using. So 4 contacts, and the stimulation you can do are limited. But now in the last 4 or 5 years, you can see all these other electrodes that have come up on the market. So more contacts, more possibility of stimulation, and now you can even steer the stimulation preferentially towards one side versus the other one by having these segmented contacts. So you can now imagine that it's quite unfeasible or impractical for the neurologist to test all of this. So now, while these technological advancements seem good at first, it also brings practical issues of what to do with it. So having a rapid tool to program the patients is pretty timely. So what about other DBS indication? As I was saying, is Parkinson is I guess, is nice in a way, because you see the patient get better right away. But what about these other disorders listed here? And that's only a few of them where there's no feedback. It takes a long time. And, this programming is pretty much just a trial and error until something works. So this would be particularly useful for these other disorders as well. So we did a similar experiment this time with psychiatric patients. So we had 17 patients in the subcallosal cingulate, so at the inferior frontal brain. And we did a very similar to Parkinson disease patients with slight methodological variations. And the first thing we did is in A, we looked at where were the most or the largest change in Bose signal. And as you can see here, it was the ACC, or anterior cingulate, and then the precuneus areas. And that was reassuring for us, because at least we did not get the motor cortex. So that was a good proof of concept that we're getting at least roughly what we were thinking we were going to get. And then what we then did is we asked the question, does the magnitude of the change in Bose signal across these 3 main areas, does this relate to clinical outcome? And you can see in the top right graph where HAM-D scale here is a scale for depression. And you can see that if you look on the x-axis as the observed or empirical, clinical improvement, then you can see that it relates with a significant correlation with what we predict based on the pattern of Bose changes in these 3 green balls. So this showed us that even in a cohort that doesn't have immediate clinical feedback, our fMRI signal still relates to clinical outcome that takes weeks or months to get. And then we did a similar experiment, but this time in dystonia patients. So now in panel A, you can see where most of the Bose signal changes was. And now we're back with motor areas, because dystonia is a motor disorder and the electrode is in the motor place in the pallidum. And you can see that the sensory motor area, the supplementary motor area show up, which is expected. And in panel B, we did a similar analysis where we could have a relationship of the Bose signal in these areas and relate it to the actual improvement. And then I'm just going to finish by mentioning, I guess that they're not super scientific because they're case reported, but I guess they're fun in a way, because before nobody would really understand what happens, and now maybe we have a glimpse of what happens. So this was a patient that had surgery, she had dystonia, and then the globus pallidus electrodes. And she was an avid dancer and swimmer. And then after the surgery, apparently she drowned almost a few times while swimming. She just couldn't swim and she couldn't dance. And so, so we asked her to do an fMRI, and we happened to also have other dystonia fMRI. And what's interesting is here in the panel A, you can see electrodes are actually, where they should be. It's not that they're in the wrong place. But then what's interesting is at the bottom row, which is row D, if you look at the bold signal and her SMA signal, it's markedly different than the other patients which do not experience this lack of coordination that's triggering her not being able to dance or swim. And so this, I think, is a true example of, why I guess this approach is better in a way than the anatomy is, two patients, same electrode position, but, somehow their SMA responds markedly differently. So the wiring is different, which is reflected here with the fMRI acquisition. And finally, there's another patient that had what's called alien limb syndrome, which is an involuntary movement of the limb, and the patient thinks it's not their own limb, it's some sort of alien limb. And this patient had the electrodes in the thalamus for tremor. And then on a certain setting, she just experienced this all the time. And even if we blinded her, didn't tell her, she would do it. So it was a true phenomenon. And then on other settings, she would respond, her tremor would get better, but this alien limb would not have it. And then if you look on the panel B, and then you can see the left one is where the alien limb was evoked, and the right one is where the alien limb was not evoked, but tremor was better. And you can see that in the left temporal parietal areas, there's different areas of both signal that happens that do not happen where tremor gets better. And what's interesting with her is she also had multiple sclerosis with a lot of plaques. And I guess one hypothesis would be that the multiple sclerosis, damage her wiring, which got rewired in a certain way that makes her experience this rare phenomenon that, and 99.999% of other people in the thalamus do not get. So there's something about her wiring that is different, and we can, see this difference with the fMRI. So I guess in conclusion, I hope I showed you that this in vivo or concurrent fMRI with PD, but with other disorder, can be a powerful tool to explore the effects of DBS and the underlying mechanism of action. But we still need to, thoroughly understand the safety to make sure nobody gets hurt, although this issue is becoming less and less, as I was explaining. And then it can act as an objective and rapid biomarker of DBS efficacy in Parkinson's, but also likely in other disorders that have delayed clinical feedback. And I guess in summary, when I was thinking about this is the retrospective approach has advantage as you can look at all the data from multiple institutions and pool it and see if you have trends. And this can improve our understanding of where to target, what circuit to target. But again, the problem is it's not all about anatomy, it's about also intrinsic brain wiring. So this has, limitation, and applying just to the single next patient, I don't think is feasible just based on that. Whereas the prospective fMRI ones, I guess it's a direct probing of brain activity that bypasses this limitation. Now, obviously there's limitation, which all the limitations that come with fMRI, namely artifact, low SNR, depending motion and all that, and also the technical challenges that are added when you scan a patient with DBS. And finally, there's probably three main programming strategies right now on the market, or pushed by different people. One of them is anatomy, and this is probably the most popular one by the vendors because it's easy to use. You put a pre-op, post-op image, show the electrode, and put your nice little VTA on the structures you want to stimulate, and that's the end of the story. But again, similar to the retrospective approach, it doesn't work for everybody. Not everyone's brain is the same. Now there's also electrophysiology, which now the newer model, what they can do is they can deliver electricity, but also record electricity. And the idea here is that based on what's being recorded, then you can make a closed loop signal that will adjust the stimulation based on what you're recording. Obviously, this comes with the advantages of being high temporal resolution, high spatial resolution, but it's expensive, it's novel, not all accessible hardware. And I guess the main limitation comparing with the fMRI is it only provides a local assessment. Whereas the fMRI, as we were talking about, provides a whole brain assessment. But again, it's not the easiest to do. It's not the cheapest thing to do either. And it has low temporal resolution compared to electrophysiology. But I guess in the end, probably the approach that will win is probably a mix of all of this, right? Like you would first see where the electrode is, have a rough idea where you need to stimulate to have a rough idea what to start with. Then you could adjust this rough stimulation with the fMRI and on an ongoing basis reassess with electrophysiology that keeps recording and adjusting the stimulation, right? So the winning strategy is probably a mix of all of them in the future. So again, I would like to acknowledge everybody at UHN that helped me and did this with me. And then, yeah, thank you for having me today. And if you have any questions.